Welcome back. The subject of this lecture is genes and chromosomes. In the last lecture, I described how the nature of genes as particles was first defined by Mendel and later shown to be true in all other organisms. Now we define the gene further in terms of its particulate nature. My opening story concerns people who weren't geneticists, weren't scientists, weren't even doctors, and figured out some genetics. And it's an old story, a very old story. In the Jewish religion, all males must be circumcised as a symbol of their covenant with their God. In the Old Testament, the first story of this was with Abraham, the founder of the Jewish religion, and was later codified in the book of Exodus with Moses. As in almost all religions, scholars spend a lot of time trying to figure out what the ancient texts mean, and the Jewish religion is no exception. There have been many commentaries written by rabbis, the Jewish teachers or ministers, about the Old Testament. One of the most remarkable is the Babylonian Talmud. The Babylonian Talmud is a commentary on the Old Testament, the 5 books of Moses, and this commentary was written in pieces from about 400 BC to about 200 AD. Now let's picture what's happening here. Here are Jews wandering around out in the desert with their Ark of their Covenant, trying to figure out what God was really telling them in these texts. And in one passage, the rabbis have a dilemma. Boys are bleeding to death when they are infants and they are circumcised. What's going on? Well, the boys have to be circumcised, otherwise they're not going to be admitted to the Jewish religion. But dying is not a good idea. What the rabbis note in this biblical commentary was that the boys who bled to death were all the sons of certain mothers. Now, in those days, you may know, women had children virtually every year during their reproductive lives. Many children would die in infancy because of infectious disease or other reasons, so they were essentially producing a lot of kids. The boys dying, as I mentioned, were all the sons of certain mothers. That is, a mother would have one boy that died, and then she might have a normal boy and a girl, and then she might, a couple of years later, have another boy that died, and records were being kept by the rabbis of this. So the rabbis decided to create some rules for circumcision in the case of the boys that were dying. That is, they looked at the pedigrees of these children. They didn't actually draw out the pedigrees. They described them in their biblical commentary. And what they described was that if a woman even remarried, she still would oddly produce some children who had this obvious disease of bleeding to death. So they decided that after a woman had more than 2 sons who bled to death, all further sons were exempt. They were the only males who were ever exempt from this circumcision rite, and it didn't matter who she was married to. It seemed to be passed through the mother. They describe in this passage of a series in which there were 2 sisters, both of whom had sons who died, and in this instance they said, if 2 sisters have sons that died, both of their future sons will be exempt from circumcision. Without knowing anything about genes and chromosomes, these rabbis, working probably 2,000 years ago, trying to figure out what God was telling them in their Bible, had described a genetic disease called hemophilia, and had described a type of inheritance, a pattern of inheritance called sex linkage or X chromosome linkage. Now today we know all about this particular disease. In fact, through biotechnology, as I'll describe later on in the course, the missing phenotype, that is the chemical that's missing in the children who have hemophilia, has been purified and manufactured by biotechnology. And now it's very unusual for someone to die of this bleeding disease. You can just give this child or an adult who has hemophilia the mystery blood clotting factor, and that's what it turns out to be. These are called the hemophilia rules. So you really don't have to be a scientist or a geneticist to figure out genetics. You could just be a theologian trying to figure out what your God is telling you. I want to talk about the particulate nature of genes now. And first I'll describe how cells carry genes and what cells are. And then we'll begin to narrow it down. Where are the genes in the cell? Well, they're in the cell nucleus. So I'll describe what the cell nucleus is and how we know the nucleus carries the genes. Finally, I'll even narrow it more to describe how chromosomes inside the nucleus carry the genes. So you see, we're going from organism to cell to nucleus to chromosome. Remember, mechanism is one of the major ideas of biology. We're not giving up saying genes are mysterious, they're part of the vital force, so we'll stop there. We're saying there is a chemical or physical explanation. We'll keep looking for it. What's a cell? Well, I described earlier on in cell theory that complex organisms are made up of cells. Remember, that's another one of the major ideas of biology, besides mechanism and evolution. And I'll define cells by looking at a human disease, an infectious disease, pneumonia. You may be familiar with what pneumonia is. Many people die of pneumonia still every year, even in hospitals. Pneumonia begins with chills and fever and a cough. It's obviously a disease of the lungs. And in that cough is, well, we'll call it phlegm. We'll call it sputum. That's a fancy word. How about that? That's the medical term, sputum, for the gunk that you cough up when you have pneumonia. So let's use a medical term and we can impress people rather than gunk. Now, how do we find out how pneumonia is transmitted? Well, it's pretty clear. We found out by looking at the pattern of transmission, and what you find is it's transmitted when one person sneezes on another. So the droplets in the air of the sputum, and some of those droplets end up being breathed in. And whatever the organism is that's infecting a person and causing the disease is in that sputum, and then it gets inside the other person, and that's how it spreads. How do we identify the causative agent? Identifying causative agents that are very small, microscopic, is the province of a field of biology called microbiology. And the founder of modern microbiology lived in the 19th century. He was a German named Robert Koch. And Robert Koch came up with a series of experiments that would provide evidence that an infective agent causes a disease. Just knowing that the infective agent is there is not enough. You have to perform a dynamic series of experiments. Biology as a science doesn't do well with correlations. Correlations is more the province of advertising, right? You'll be happy if you have this car. Happiness and car go together. Or politics. The economy is bad because of this politician. That's not necessarily cause and effect. That's two events happening at the same time. That's a correlation. Just because an infective agent is present with the disease doesn't necessarily indicate that it causes the disease. By cause, we have to do science, and science means experiment. And Robert Koch proposed his postulates or experimental scheme. And his experimental scheme is in 4 parts: isolate, infect, symptoms, and repeat. Isolate means you go into the sputum and you isolate your suspected organism, whatever it is. It might be a bacteria or a protozoan or some other organism. Isolate. Then infect. Now what you do now is do an experiment where you infect some creature. Now in humans, in human diseases, we can't infect a human with what we suspect to be a disease-causing agent. That's unethical science. We would never think of doing that. So we use animal models, and this is where animal research turns out to be extremely important. It's the only way to find out if an agent is infective. And if that agent will infect an animal, we have to make sure that it's possible. Then we allow the infection to proceed, and now we look for the third aspect of Koch. Remember, isolate, infect, symptoms. We look for the same symptoms, in this case pneumonia, in the animal, and see that the animal has a disease similar to the human disease. And finally, repeat, which means we re-isolate the suspected disease-causing agent from the animal, and we repeat with the second animal. Isolate, infect, symptoms, and repeat. And this series of experiments has gone on for infective agents since Robert Koch's time. If you look at pneumonia, there are two different kinds of organisms that we can isolate, and they behave, and their chemistry and their biology are very different, yet they both cause pneumonia. The first type of organism is very small. You can't see it with the naked eye. In fact, it's a rod-shaped or spherical object about 1 micrometer in size. And what's a micro? A micrometer is a millionth of a meter, or a thousandth of a millimeter across. Now, a thousandth of a millimeter, I put my two fingers together, the closest you can see those two fingers apart is about 1/2 of a millimeter. That's about the limit of the human eye to see. So we can't see things that are smaller. Well, this agent is smaller than that, so you have to use a microscope to see it. If you take this agent and you prove that it causes pneumonia by Koch's experimental series, you can grow it in the laboratory if you supply it with sugars and water and things for it to grow, and it'll grow and reproduce very quickly. If you look at its chemistry, it's very complex. It's got thousands of different chemicals inside of it, water being the majority, but thousands of other chemicals as well. This is a bacterium. The name of the bacterium is pneumococcus. And the bacterium will grow in a laboratory setting. The second type of agent also causes pneumonia, just like Koch's postulates, goes through all the experimental series, isolate, infect, symptoms, and then repeat. The second agent is much smaller than the agent that causes pneumonia. It's about 1% the size. You can't even see that through a regular microscope with lenses. You've got to use a fancy microscope called an electron microscope that magnifies even more. This thing is very different from the bacteria. It's not only smaller, but it can't grow and reproduce on its own in a laboratory environment. It needs other tissues to grow. It's a parasite in its growth characteristics, and it's chemically simple. It's a virus. In this case, the virus is called respiratory syncytial virus. Bacteria, they're complex, exhibit all the characteristics of living things, are cells. Viruses, much simpler, are not cells. Cell theory tells us cells make up living things. In the case of bacteria, they are living things. Cells exhibit all the characteristics of living things. For example, some of these characteristics are that they're complex. In fact, viruses, of course, are very, very simple. Living things have many chemical reactions that go on in them. Living things have the ability to grow and reproduce. Living things can determine what comes in and out. They have a boundary between them and the environment that determines what things come in and out of the cells. Non-living things can't do all of these things. You know, snowballs can grow. Snowballs can reproduce, right? But snowballs can't refashion things inside. They don't have these complex chemical reactions. And snowballs can't say, well, I'm going to let some snow in or not let snow in. They don't have this ability to determine what comes in and out. So cells have all of these characteristics. There are two types of cells in nature. We recognize prokaryotic cells. The term karyon does not mean baggage. It means a body. It's a Greek term that means the body. Prokaryon means before the body. It's a cell that is relatively simple. It doesn't have complex bodies inside of it. So bacteria are examples of a prokaryotic cell. You'll know what I mean when I talk about the other type, which are the eukaryotic, eukaryotic, the true cells of complex organisms, like cells in us, they all have complex internal substructures, including the most obvious cell structure, which is the cell nucleus. And when you look at a cell under the microscope, the cell nucleus is by far the most important and prominent part of the cell. The cell nucleus is where the genes reside. Now, how do we know this? We know it through experiments, not by just saying that's where the genes are, but by proving that's where the genes are by experiment. Let's do a couple of experiments on the cell nucleus. One of the cells that's a favorite of high school biology and grade school biology is the amoeba. It's this blob-shaped thing. It's pretty big, a couple of 100 micrometers in size, and it kind of flows around on a microscope slide, and you see a nucleus inside the amoeba. And because it's so big, we can do the following experiment. We take a glorified straw. Now in science, we don't use the term straw, we use the term pipette. It's a glass straw. Okay, it's a straw. We suck out the nucleus. So we puncture the boundary of the cell and we suck out the nucleus. And we're going to see what happens. Now, of course, we have to compare that to a cell that is equally treated, but you don't suck out the nucleus. So we do what's called a sham or fake operation, where we puncture the cell, but we don't suck out the nucleus. So we do exactly the same things, but we don't suck out the nucleus. Well, what happens? The cell with the nucleus survives, the cell without the nucleus dies. Conclusion, the nucleus is vital to the survival of the cell. Please send Nobel Prize to the following address. That doesn't tell us much about genetics. Well, we can start looking at genetics and we look at cells that have a little bit more complexity. There's a plant called acetabularia that lives in the intertidal zone, Pacific Northwest through Western Canada. And this cell has specialized parts to it. And the specialized part is on the top of the cell. It adheres to a rock on the bottom of the cell with a nucleus, and the specialized part is on the top. One of the amazing things about this organism is you can cut it off, and if you cut off the top, it regenerates the specialized part. Now, there are different species. One of these species has a top that looks like an umbrella, another has a top that looks like a flower. So they're different shaped tops. A series of experiments has been done that shows that it's the nucleus part that determines what the top will be. If we cut off the top from the cell and the nucleus is still there, it will regenerate a top, but it's always species specific. So now we know the cell nucleus determines genetically the inheritance of this particular organism, right? What type of top it will make. What about complex organisms, like a carrot? A carrot has a root, a stem, a leaf, a flower. In the root is different tissues, etc. The part of the carrot we eat is the carrot root. And it has different types of tissues inside of it. Just like your arm has different tissues, right? It's got muscle and bone, etc. So the carrot root has different tissues. In 1958, Frederick Stewart at Cornell University took a specialized cell from the root of a carrot and asked the following question. Does the nucleus of that specialized cell still have all the genes for the rest of the cells of the organism? We know that the fertilized egg of the carrot is totipotent, meaning it has the potential to be everything, to be toti. In other words, it has all of genetic information to form the root, the stem, and the leaf, and the flower. What about when a cell specializes? Does this nucleus still have that genetic information? Stewart did the following experiment. And it's a very historic experiment, even though it's almost 50 years old. He took the root of a carrot plant, took some cells out of it, put them in a defined chemical medium, and lo and behold, the chemical medium that he defined, which was an embryo extract, told this root cell to stop being a root cell, but start being an embryo. It reverted to where it was before, and it divided and formed a carrot embryo and formed an entire carrot plant that would reproduce. If you took 10 cells from the carrot, you got 10 identical plants. These cells are cloned. A clone is an identical group of cells or organisms that is genetically the same as the original progenitor of that group of cells or organisms. And here we could have 10 carrot plants that came from a single carrot. Now, the cloning aspect is something we'll come back to later on in the course. The genetic aspect is what I want to emphasize here, and that is that the carrot root cell hasn't lost any of its genetic capabilities. It has all of the genes to give a whole carrot. Now, the gene in the nucleus is carried on large visible structures called chromosomes. And I want to turn to them now as our quest to further define the gene goes on and gets down to a finer physical level. Chromosomes provide an explanation for Mendel's laws of genetics. Remember what Mendel said? Mendel said that every organism has 2 copies of every gene, every elementon. One from mom and one from dad, one from the male, one from the female. By the same token, every organism has 2 copies of every chromosome. Genes are located on long strands of genetic material that are packaged into compact structures called chromosomes. We can see the chromosomes when cells reproduce. Now, when cells reproduce, all of the genes have to duplicate. Because if I have 10 genes, 5 from mom and 5 from dad, let's say, and I want to produce 2 new cells from one cell, if there are 10 genes in that cell, I have to have 10 genes in each of the new cells. So that implies that before reproduction happens, before these genes are separated, I have to duplicate them. And that's exactly what happens. There's this dance of the chromosomes in this beautiful process called cell division. It's really cell reproduction, in which the chromosomes first duplicate, and then 1 copy of each chromosome goes to the ends of the cell, and then 2 nuclei form. And the important thing is these 2 nuclei in the new cell contain, each of them, a complete set of all the chromosomes. So the chromosomes had to duplicate before. In humans, there are 23 pairs of chromosomes. For each pair, of course, we inherit 1 from mom and 1 from dad. Our gametes, or sex cells, eggs or sperm, contain 1 copy of each of these chromosomes. So a gamete will have only 23 chromosomes and not 23 pairs. Specific genes have been linked to specific chromosomes. And since we have undertaken genome sequencing, we know with quite a degree of specificity where genes are on which chromosomes. As I mentioned, because humans have 24,000 genes and 23 pairs of chromosomes, we have about 1,000 genes per chromosome. Think of the genome, the entire genetic complement, the sum total of all the genes, as a library, and the chromosomes as volumes, and the genes as paragraphs. That's a good way to look at it. Now, in mammals, which includes us, there's a special set of chromosomes, and these special set of chromosomes are the X and Y chromosomes. The X and Y are rather different from each other. The X is a large chromosome, typical of most chromosomes, and it contains several thousand genes. The Y chromosome contains very few genes, even though it's part of the pair with the X. People with Y chromosomes are males. People without Y chromosomes are females. Now, everyone has X chromosomes because X chromosomes contain many genes that are essential to the survival of a human or other mammal. It just happens to be one of those volumes in the encyclopedia. But it's the Y chromosome uniquely that contains the male determining gene. Now, first of all, how do we know that the Y chromosome is necessary for maleness? Well, that's not too hard. We know it from clinical cases. Some people inherit more chromosomes through some abnormalities in cell division in forming gametes from their parents. So, for example, there are people who have not an X and a Y, but instead have an X and a Y, 2 X's and a Y. So these people have 23 pairs of chromosomes and 1 more, 47 chromosomes. The XXY the person is a male, even though they have two X's. There are people who have XXXY, male. There are other people who have 45 chromosomes and just an X. Now, you have to have at least one copy of every chromosome. You're not going to be missing 1,000 genes and survive. And if they have one X chromosome with no other chromosome along with it, they have, of course, 45 chromosomes, 22 pairs plus the X. These people are female. So it's the Y chromosome that determines maleness. In fact, in the embryo, we all start out looking like females in our anatomy very early on in the human embryo. And if the Y chromosome is present, that female plumbing kind of is degenerated and the male takes over. It's an interesting way to look at life, to say we all start out in some ways looking like women. What are the genes on the X chromosome? We know the genes on the Y for maleness. The genes on the X chromosome include the gene involved in blood clotting that is defective in hemophilia, which I described earlier on in the opening story. Another gene on the X chromosome is a gene which, when defective, leads to red-green color blindness. Both of these are recessive. And they are both much more common in males and females. Why? Well, again, think of a male. A male has a Y chromosome that doesn't have the genes that are on the X. So any gene in the X, dominant or recessive, will be expressed in males. Whereas if a female inherits, for example, the recessive allele that would lead to hemophilia, that woman will not express that recessive allele if she also has the dominant allele on her other X. So that's why X-linked abnormalities are much more common in males than females. Sex determination, male or female, forming sperm or eggs, is called primary sex. And to show you how genetics works, scientists have recently been able to narrow down, even on the X chromosome, the location and identity of the sex-determining region. The sex-determining region on the Y chromosome is called, not surprisingly, SRY. Listen, SRY, well, S, sex-determining, R, region, Y on the Y chromosome. Now, we know it's there somewhere. How are we going to find out where it is? Well, take the case of a girl who had an X and a Y chromosome. And the doctor went, "What? You have a Y chromosome. You must be a boy. How could you be a girl and have an X and a Y chromosome?" When they looked carefully, they found a very small piece of the Y chromosome was missing. And they figured, oh, that's the region that causes maleness. It must be missing. Take the case of a boy who is XX. 46 chromosomes, 2 X's, no Y. When they looked carefully at his chromosomes, they found an additional piece of genetic material on one of his chromosomes turned out to be, you guessed it, that same piece that was missing in that girl, the sex-determining region on the Y chromosome. The lesson in this, and the reason I bring it up, is that these clinical cases tell us cause and effect in biology. By looking at what goes wrong, we can infer in the normal situation and what is right. So science can do experiments in this way, not actually manipulating themselves, the chromosomes, but by looking at people who are born with already defective chromosomes in some way. Secondary sex, the outward appearance of us, the deep voice of men and the hair on the chest, or the higher voice on women and breast development, etc., is determined by hormones. It's not determined by the X and the Y chromosomes. The hormones will be familiar to you, estrogen and in women and testosterone in men. In this lecture, I've tried to describe the particulate nature of genes all the way down now to a specific region on a chromosome. Now we turn to the chemical identification of the genetic material, and that is DNA, in the next lecture. Thank you. changes the bacteria into a bacteria cell to a viral factory. The generality of this: DNA is the genetic material not just of cells like bacteria, but even of particles that sometimes are alive like viruses. Attention now turned to the nature of DNA itself, and that's the subject of our next lecture. Thank you. We found it inside the nucleus as DNA, and I described experimental evidence in several different systems for DNA as the gene. Now let's see what DNA really is and how its structure fulfills the requirements we have for the genetic material. My opening story concerns the Iceman. In 1991, a group of hikers in the Alps between Austria and Italy spotted a corpse frozen in the ice. There were some tools and a dagger nearby. The body was carefully removed, and 4 days later, the body, now known as the Iceman, was at the University of Innsbruck in Austria. Scientists determined that this body was 5,000 years old. It was a male and had been killed by an arrow, so he was a victim of homicide, and then somehow froze into the ice. There are many remarkable things about the Iceman, but to me the most remarkable is his DNA was preserved in his bones, virtually intact. When scientists looked at the information content of his DNA, they found that this information content placed him smack in the genetic group of the current Europeans who live in the alpine regions of Europe. So this is one of the forefathers of the people who currently live there. That DNA can last so long is testimony to its durability. It's nice to have a genetic material that will be stable and not break down very readily. The structure of DNA is testimony to something else. I call DNA a secular icon in our society. Now, what's an icon? An icon is an image or symbolic representation that is often used in sacred situations. In this case, it's secular. The 20th century had two great icons that symbolized science in the public mind. One is the atom with the whizzing electrons going around a nucleus. The other is DNA with this double helix. People now talk about DNA all the time as a shorthand. An advertisement for a financial services company tells customers, "We understand the DNA of business." A perfume that I actually tried, didn't go over big, is called DNA. And on the bottle it says, "This is the essence of life." Didn't enliven me up very much at all. There's a media software system called the DNA server. There's a clothing line called DNA Divewear, and it advertises that this is the genetic design extreme behavior. And of course, there's the original biotechnology company whose stock market symbol is, you guessed it, DNA. DNA is the subject of artists as well. Salvador Dali used the DNA double helix in some of his art. There is a portrait in England of the Nobel laureate Sir John Sulston, who worked on cell cycling, cell division in yeast cells. And what it has is parts of his DNA inside of bacterial cells in the portrait. A Brazilian artist named Eduardo Koch used the chemical building blocks of DNA to translate verses of the Bible. And viewers looking at this work of art can change the DNA and change the verses that it represents at will. I've seen playgrounds with slides shaped in the helical form of DNA. And of course, it's hard to visit a pharmaceutical company or biotechnology laboratory that doesn't have a DNA sculpture somewhere. So DNA really is everywhere in our society these days. Now, I want to talk about DNA structure in terms of its function as genetic material in three ways. And first, I'll describe the early studies that suggested its structure. And I'll take you through how we determine what DNA was structurally. Then I'll describe some of the properties of DNA as a double helix. And finally, I'll describe how this double helix structure of DNA fulfills the requirements of genetic material that I talked about in the last lecture. First, the early studies of DNA structure. I mentioned in the last lecture that Friedrich Miescher was the first person to isolate DNA from cell nuclei. Miescher is a very interesting man. He was the son of a doctor and graduated from medical school at the top of his class at the University of Basel. Unfortunately, Miescher was partially deaf and decided for this reason not to pursue clinical medicine. Today, many physicians who have handicaps are in medicine. In those days, it was wise not to do so. So instead, Miescher chose a career in research. He began working on the chemistry of the cell nucleus, about which people knew nothing. His mentor was a world-famous, at that time, German biochemist named Ernst Felix Hauptseiler. In fact, Hauptseiler is so famous, there's a journal called Hauptseiler's Journal of Biochemistry, which until recently was called that name. Now it has a different name. For some reason, they wanted a shorter title, maybe for indexing purposes. Working in Hauptseiler's laboratory, Miescher in 1868 isolated nuclei from the cells of blood that was on dressings at a nearby military hospital. So from these dressings, he got white blood cells, isolated their nuclei, and then from the nuclei, he busted them open and extracted a substance that was rich in the usual things that you find in biological molecules: carbon, hydrogen, oxygen. But two other elements were there: phosphorus and nitrogen. Because this substance was in the nucleus, he called it nuclein. One of his students later called it nucleic acid. And later on, when we found out what it was made of course, it turned out to be DNA. Now, Miescher's boss, Hauptseiler, was so amazed at this new substance. It formed these long fibers. DNA is not that hard to isolate. I've done it with grade school students. It's so easy. These long fibers impressed the boss, Hauptseiler, so much that before he let Miescher publish his work, he went into the lab personally and repeated all of Miescher's experiments. This was so novel, Hauptseiler wanted to make darn sure that this stuff is for real. Well, he repeated all of Miescher's work and then allowed Miescher to publish the research. Nucleic acid was a curiosity, even though Hauptseiler had published it as something in the nucleus, until it was realized sometime in the next 30 years after this, remember this is the mid to late 1800s, that the genetic material is in the nucleus. You remember those experiments I described of nuclear transplants and of regenerating cells, and that genetic material is in the nucleus and that's species specific. So right away, when people found DNA in the nucleus, they said, this is a candidate for the genetic material. But I mentioned previously that there's not just DNA inside the nucleus, there are proteins as well. So people wanted to find out, well, which one is it? Now we know the answer in advance, but I want to take you through some of the reasoning that led to that answer, especially the chemical reasoning, because it's important that we understand the chemistry of DNA, not down to the last atom, but in terms of its information content and what it can do in terms of genetics. In the 1890s, a chemist, Albrecht Kussel, found that DNA was a polymer. Now there's a new word for you. A polymer can be considered like beads on a chain. The long chain is the polymer. The individual beads that make up the chain are monomers. So monomer and polymer. And that DNA fibers that Miescher had isolated were these long polymers of probably millions of units of monomers. And the monomers were the things that had nitrogen in them, as well as carbon and hydrogen and oxygen. And there were 4 what are called nitrogen bases that made up DNA. These are bases, as the spelling in the bases on a baseball diamond, and they're called adenine, thymine, guanine, and cytosine. Now, we abbreviate them A, T, G, and C. Those are the different monomers that make up DNA. So if we're talking about a long chain, we only have 4 types of beads in it. Red, orange, yellow, and green, let's say, right? And so our chain is made up of information that contains red, orange, yellow, and green. And so the chain might be red, orange, yellow, green, red, green, orange, yellow, red. I'm not going to continue on that. There can be very many different orders of the beads on the chain in different amounts. Let me give you the names of the 4 of them again, and then I will not mention the names again in the course. I'll just call them A, adenine, T, thymine, G, guanine, and C, cytosine. Students often are so used to the shorthand of A, T, G, and C, they think they're part of the alphabet and they don't stand for substances, complex molecules in themselves. So I wanted you to give them a name. Between 1900 and 1930, working at the Rockefeller Institute in New York, a nuclear chemist named Phoebus Levine spent 30 years studying DNA. Levine found that the 4 monomers, those bases A, T, G, and C, were not linked to each other in DNA. Instead, they were attached to a backbone, and the backbone was made up of a sugar and a phosphate group. Phosphate is phosphorus with oxygens attached, and the sugar is a multi-carbon molecule that has 5 carbons and 10 oxygens and 10 hydrogens and 5 oxygens. The sugar is called deoxyribose, and so the name deoxyribonucleic acid. Deoxyribose is the sugar. Nucleic acid is the name that Miescher's student gave to this stuff. Deoxyribonucleic acid was coined for this stuff by Levine, and it was called, from then on, DNA. Levine, again, showed that the bases A, T, G, and C are not directly hooked onto each other, but are hooked onto this boring backbone of sugar and phosphate. So it's kind of like a chain that is a gold chain that has little subchains hanging from it, and then the beads are on the subchains. And the gold chain can be the sugar-phosphate backbone, and then the beads hanging off of those individual chains are the bases, A, T, G, and C. Now, Levine proposed that DNA was indeed a very, 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 very long polymer, and it consisted of repeating units. And he said the repeating unit was ATGC. So we could define a DNA molecule as ATGC, 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 and as I ask my students, can I stop now? And some of them say yes, and thank God then I stop. If they don't say yes, I keep going for the hour. So it was just a repeating unit of ATGC according to Levine. If Levine was correct, remember this is about 1930, and Levine certainly thought he was correct, this meant that DNA lacked the variable information content to be the genetic material. Something as boring as that, same unit repeated many times, that's not going to give 24,000 genes or even 1,000 genes. Levine was a towering intellect, a stupendous chemist, a strong personality. So people believed him. I like to think in terms of nucleic acid, Levine is the Aristotle of the early 20th century. So everybody said, well, that's it for DNA. That's not going to be genetic material. Levine said it's just repeating units of ATGC. So people started looking at proteins for the genetic material. Now, this is the 1930s, and remember, DNA was not identified experimentally until the mid-40s as a genetic material in the bacteria transformation experiment. In the late 1940s, another great biochemist took a stab at DNA by the name of Erwin Chargaff, working at Columbia University. Chargaff found that Levine was wrong about the polymer of DNA. It wasn't ATGC at all. It was highly variable. So if our DNA starts with ATGC, Chargaff said, oh no, it's ATGCTTCAA. Did you get that exactly? GCTTA. It was highly variable. A very important finding because right away people said, whoa, there's a terrific information content here. Chargaff made other discoveries whose significance came to be fully appreciated shortly afterwards. First of all, for each species he examined, when he chopped up the DNA into individual beads, monomers, he found a constant ratio of the proportion of bases. So for every human being, there was a certain percentage of A that was the same, a certain percentage of T, a certain percentage of G, and a certain percentage of C. They added up to 100%. For each eucalyptus plant, there was a different percentage of A, but T, G, and C. The second thing chemically that Chargaff found was that the ratios of A and T and G and C were the same. That is, if the species had 20% A, there would be about 20% T. Therefore, we're going to have to 100%, 30% G and 30% C. So A was always equal to T in proportions, and G was always equal to C. Another species, another creature, might have 18% A and 18% T. And of course, 32% G and 32% C. And now here's your homework assignment. 15% A, tell me the T, G, and C. Hand in the answer tomorrow, send it to the teaching company. Now the problem was fitting all of these observations together. Physical chemists came into the picture now. Not biochemists who look at what things are, but physical chemists who look at the arrangement of atoms in three-dimensional space. Consider water. Water is H2O. There's an O with a couple of H's attached. It's very regularly shaped. All water molecules are the same. Now consider DNA. Millions of atoms. Was there a regular shape to DNA? Well, there's a physical way to examine DNA, and that's by shining X-rays on the DNA molecule when it's in a solid form. Now, when X-ray is shown on a DNA molecule in a solid form, what happens is the rays go into DNA and then they get bounced around by all the atoms that are there. And then they come out the other end. Now, if we put a photographic plate at the other end, we're going to get a pattern depending on how much bouncing around that ray has gone through. So the ray goes through the DNA molecule, then it bounces. And you get these what are called X-ray patterns. And the X-ray pattern of DNA was very regular. Two scientists working at King's College London in the early 1950s, Rosalind Franklin and Maurice Wilkins, did X-ray analyses of DNA and found a regularity that they interpreted to be in the shape of a helix, a coil. So now we had a lot of evidence for the shape of DNA as a helix. We had a sugar phosphate backbone that Levine had found. We had A equals T and G equals C that Chargaff had found. Working at Cambridge University, up the road from London, a young American geneticist, James Watson, and a somewhat older British physicist, Francis Crick, looked at all of this evidence and came up with an inspiring idea. Maybe, just maybe, DNA is a double helix, that there are two polymers here, and that the sugar and phosphate are on the outside, the regular sugar phosphate, and the bases are on the inside. And to show that this might be true, Watson and Crick made ball and stick models of the bases of DNA and the sugar and phosphate and tried to fit them together tightly. And the only way they would fit together as a double helix was by having the sugar and phosphate on the outside and the bases on the inside, and the A's and T's just fit together. And the G's and C's just fit together like a ball into a baseball mitt. Now they fit. They didn't bond very tightly. They fit very weakly together, but they fit. No other type of structure worked than the double helix. Crick's wife was an artist, and she drew a double helical shape with the bases on the inside and the ribbon on the outside being the sugar and phosphate for the article they published in the spring of 1953. It's a one-page brief article describing the evidence and their proposal for the double helix model. This model was immediately accepted as the model for the genetic material, not only for its science, but for its elegance. It's a beautiful structure. And of course, this brief one-page paper won them the Nobel Prize. Nobel Prize is given for people who think, not necessarily for people who do a lot of grunt work in the laboratory. The DNA double helix fulfills the requirements for the genetic material. Now, what are those requirements? There are 4 of them. First, DNA should have a huge information content. Second, DNA can be accurately replicated. Third, DNA should be expressed as the phenotype. And fourth, you should be able to mutate DNA, to change it and get genetic mutation. I'm going to deal with the 3 of the 4 now, and I will defer to the next lecture the aspect of expressing DNA as the phenotype. First, the information content of DNA. Well, this really came from Chargaff, as I mentioned. DNA has a huge information content. With just 4 bases in any amounts, we can make all types of words, right? It's a language that has a great number of possibilities. So, for example, there are 469 base pairs of DNA in the insulin. Insulin is a hormone, and the gene that codes for insulin is 469 base pairs of DNA. Now, when we draw the structure of a DNA molecule, we go A, T, G, C, and I'm going to read out the beginning of the insulin gene. I'm reading you a human gene information content, A, G, C, 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 T, C, C, A, G, G, A, C. We assume the other strand is there. Now, how do I know what the other strand is? Everywhere there's an A, there's a T. A and T go together. Everywhere there's a G, there's a C. Right? So we know one strand, we know the other strand. Everywhere there's an A, there's a T in the other strand. Everywhere there's a G, there's a C in the other strand. Everywhere there's a C, there's a G in the other strand. Everywhere there's a T, there's an A on the other strand. So if we know one strand, we know the other. So generally, when biologists look at DNA sequences, they look at only one of the 2 strands of DNA. There's a huge information content. Humans have over 3 billion base pairs of DNA. So there's a lot of information there. We can get lots of different sequences out of DNA, the 4 bases. DNA replication. How is DNA duplicated? One of the great coy statements in the history of modern biology was the statement that Watson and Crick made in their paper where they unveiled the double helix model. They said, it has not escaped our notice that the structure we propose will provide a mechanism for accurately replicating DNA, genetic material. What was not escaping their notice was that the double strands of DNA could unwind very easily. The base pairs of DNA, that is the A and the T, that are inside the double helix, are weakly attached to each other. And so we can envision the two strands of DNA unzipping. So the two strands separate, and each one of the old strands can act as a template for the new strands. Let me give you an illustration of that. Let us say we have a DNA molecule, I'll make it really simple, that is AAAAA. The other strand of DNA, of course, will be TTTTTT, 5 of them, okay? 5 T's to 5 A's. The strands unzip. Now on one side we have the AAAAA. On the other side we have the TTTTT. And now there should be an enzyme or a mechanism coming in to duplicate this, and the AAAA here gets bonded to TTTTT. So now we have an AT double helix. And the TTTTT here gets bonded to AAA. And so we have 2 new helices just by unwinding and replicating in that way. This is called semi-conservative replication, not because of the politics of the people who propose the model, but because you are semi-conservative. You are conserving both old strands of DNA, and you're adding the complementary or the opposite strand to the old strands of DNA. It's not like doing a Xerox copy. In a Xerox copy, we put the old copy on the machine and we get a brand new copy, right? So we have 2 copies, one old and one brand new. That would be conservative replication. It's not like that at all. It's semi-conservative, where DNA is retaining the old strand for the new strand. And there's a very interesting question we can ask about this, and that is the following. I am the product of a single cell, the fertilized egg. It had DNA in it from my mother and father. When that cell reproduced, each new cell, semi-conservative replication, had an old strand of DNA and a new strand of DNA. So in the early embryo, the first cell had both original strands of DNA, parental strands from mom and dad, and when it divided, reproduced, each new cell had one old strand and one new strand. When those new cells replicated, each one of those cells ended up with an old strand and a new strand, one new cell. And the other cell has, of course, 2 new strands because its second strand of DNA is new. So is it possible that in my body still is my parent's DNA after all of those cell divisions? Answer, I guess it's possible. I haven't seen evidence that it's there. I haven't seen evidence that it isn't. But it would be nice to think that somewhere in my body might be a cell that contains the actual DNA from my parents and those 60 trillion cells. My third requirement for DNA, the other 2 that I've talked about are information content and accurate replication, is DNA mutation or changes that are permanent and passed on to the next generation. So we can envision a normal DNA sequence might be A, T, C, G, G, T, T, A, and you change it. Instead of A, T, C, G, it's A, T, C, A or something. You change one base pair in DNA. It turns out that the duplication mechanism of DNA is error-prone. Errors do happen. About 1 in a million times an error is made. Now, if you're replicating a billion building blocks, you're going to change 1 in a million. Most of these are repaired in DNA. So DNA is constantly being scanned by a system of DNA repair mechanisms that will remove the bad base and put in the good base. Now, how do we know a bad base? Well, if you put in a C opposite an A, normally you're supposed to put in a T opposite an A. Put in a C opposite an A, that's bad. There is actually scanning going on all the time along your DNA to repair those errors. Any errors that are not caught, of course, are passed on to the next cell and the next generation. This is the raw material for evolution and for genetic diversity. The fourth requirement for DNA as genetic material is the expression as the phenotype. That's the topic of my next lecture. Thank you.